Hello friends, I'm Dr. Sanjay Gaikwad. Today we are continuing the value added course Applications of Chemistry in Industry Part 2nd. This course is taken in collaboration with the Department of Chemistry of our college and Pharmaceutical Institute of Quality Management. From this institute, Mr. Santosh Kadam, Mr. Govind Deshpande helped me to complete this course for your class. In this course, in the first part, we discuss about the general reagents, some name of the reagents, volumetric reagents and solutions, how these solutions are prepared, what type of concentrations we are measuring, how they are related with each other. Then standard solutions, they are available in the laboratory, primary standard, secondary standard, tertiary standard. Then buffer solutions, the definition, how these buffer for the different pH we prepared, and some reference materials. Then we learn some techniques like infrared spectrophotometry, ultraviolet visible absorption spectrophotometry, then the most of the chromatographic separation technique used in the industries, particularly in the pharmaceutical industry, and some qualitative reactions and test. In the second part, we are going to discuss what are the basic fundamental tests we should know or we should aware about these tests, in which some disintegration test for tablets and capsules, disintegration test for suppositories and pessaries, some dissolution technique, dissolution test for tablets and capsules, then consistency of formulated preparations according to the pharmacopoeia, under which the uniformity of weight of delivered doses from multi-dose, then containers, uniformity of content like uniformity of dosage units, extractable volume of parenteral preparations, consistency of formulated preparations. Generally in pharmaceutical industry, this is the framework. In industry, the board of management, board of director, managing director, then managers, vice managers, and the units like management, maintenance, planning, production, quality management. Our interest, that is our chemist has an interest in the quality management and plant work. Under the quality management, quality assurance, quality control, research and development, laboratories, and actually all these research we are applying on the plant for the bulk material. Then in which the first test is a disintegration test. This test determines whether the dosage forms such as tablets, capsules, suppositories. Now all these are disintegrate within the prescribed time. That is most important when placed in a liquid medium under the prescribed experimental conditions like temperature, because external condition is maintained because these are the doses used for the human beings, and the human being temperature is most important. At this temperature, how much they taken time to disintegrate the material, disintegrate the dosage like tablets and capsules. That is the first test. Then disintegration test for suppositories and pessaries. Now this test determine whether these drugs soften or disintegrate within the prescribed time when placed in a liquid in the experimental conditions. These tests are in the accordance with the latest specifications laid down in the pharmacopoeia like USP, EP, and its compliance. Now dissolution is another test, most important. We know that dissolution is the process when the solid state, liquid state material dissolves completely into the solvent. That means either ionization, either dissociation, either association, or complex formation, out of which these drugs, how they dissolve into the particular solvent. That is most important. And accordance with the pharmacopoeia, the procedures laid down in the pharmacopoeia, we can see or check the dissolution test. Now, in the pharmaceutical industry, drug dissolve or dissolution testing is routinely used to provide critical in vitro drug release information for both quality control purposes, that is to assess batch to batch consistency of solid oral dosage forms such as the tablets and drugs development, that is to predict in vitro in vivo drug release profiles. Now, difference between the disintegration and dissolution. Disintegration is a process of breaking down a substance into the tiny particles or fragments to improve its solubility in the solvent. Disintegration is also defined as that state in which no residue of the unit under test remain on the screen of the apparatus, or if a residue remains, it consists of fragments of disintegrated part of the tablet component, parts such as insoluble coating of the tablets or the capsule shell, or if any melted fatty substances present in the tablet or in the drug. This is the disintegration. While dissolution is a process through which solute dissolves in a solvent, dissolution is also used predominantly in pharmaceutical industries to check how soluble a drug in the body. Whereas dissolution is a process by which the solid substances enters in the solvent to yield a solution. It is controlled by the affinity between the solid substance and the solvent, that is association, ionization, complex formation, or dissociation of the compound, and how much time required to dissolve completely, and that is our aim. Now, the pharmacopoeia, we are using the pharmacopoeia, Indian pharmacopoeia, British pharmacopoeia, European pharmacopoeia for the standard preparation, formulation and preparation or procedures laid down in all these pharmacopoeias. Now, in the pharmacopoeia, it contains the list of drugs and other related substances. It contains the sources, that is procedures, from where we are getting the compound, the raw material, then descriptions of these tests, then formulas and preparation actions, that is the procedures, uses, doses, storage conditions, and everything about the product, the intermediate compound, then raw material, and what are the tests given by these as quality. That is, with the help of the thin layer chromatography or chromatographic test, we have to check the quality of for each part when it is transferred to the quality control department, R&D department, as well as on the plant. All these procedures should be followed, laid down by the pharmacopoeias. Now, consistency of formulated preparations. Under this, the uniformity of the weight. For example, in case of the tablet, randomly the weight of the 20 tablet or unit taken determine the average weight of each unit. Then randomly taken the weight of the any two unit, and the average and individual weight should not deviate more than percentage deviation given as per the pharmacopoeia. In case of the capsules, the weight and intact capsules, remove the weight, the all contents in the capsule, then weigh the shell. If shell is soft, then use a proper solvent to remove the contents. Then the mass of the content is the difference between the weighing, and repeat the procedure for another 19 capsules. If it is given or permissible by the pharmacopoeia procedure, then the batch is acceptable. In case of the capsule containing the solid powder material, now weigh an intact capsule, then remove the weigh the all powder contents, weigh the shell, the mass of the content is the difference between the weighing, and repeat this procedure for the 19 another capsules. Now uniformity of the weight of delivered doses from the multi-dose container. Now in case of the capsule, multi-doses are kept into the capsule. Now weigh individually 20 doses taken at random, then determine the individual and average mass. Not more than 2 of the individual deviates from the average mass by more than 10%. Then the next, consistency of the formulated preparations, under which we are learning, we are going to learn the uniformity of content, uniformity of the dose unit, extractable volume of parental preparation, consistency of formulated preparations. Now uniformity of the content. It is a pharmaceutical analysis parameter for the quality control of capsules or tablets. Multiple capsules or tablets are selected at random and a suitable analytical method is applied to assess the individual content of the active ingredient in each capsule of the tablet. In case of the capsules containing the multi-doses, now dosage unit are defined as the dosage forms containing a single dose or a part of dose of an active substance in each dosage unit. It is the degree of uniformity in the amount of the active substances among dosage unit. Now extractable volume of parental preparations like the liquids or solutions containing the drug kept into the container and given to the patient through the syringe. Now each container of an injection is filled with sufficient excess of the labeled size or that volume which is to be withdrawn. Then suspensions and emulsions must be shaken before withdrawal of the content and before the determination of the density. Oily and viscous preparations may be warmed according to the instruction on the label if necessary and thoroughly shaken immediately before removing the content, and the content are then cooled at 20 to 25 degrees Celsius before measuring the volume. Now, under the consistency of the formulated preparation, the formulation, different chemical substances including the active drugs are combined to produce a final medical product. Now there are two types of medication products like oral formulation and topical medications. Under the oral formulation, we are taking the tablets, capsules, and in topical medications, the creams, then powders, then ointments, these are included in the topical medications. Now in the process, quality control test, it is given as under the name of IPQC. Now these are the basic tests given as a hardness, friability, thickness, disintegration time, weight variation, content uniformity, dissolution test, leakage testing for strip, and blaster packaging. Now from this, the 4, 5, 6, 7, that is disintegration time, weight variation, content uniformity, dissolution, we just now seen. But in case of the hardness, now hardness for IPQC, it is the load required to crush the tablet when placed on the edge, and some pressure is applied, and then we are measuring the pressure, and under this we are measuring the hardness of the tablet. It affects the disintegration because more harder tablets, they require the disintegration time is more, and therefore it affects on the disintegration. Higher hardness and disintegration time within the time, then accept this batch. Now friability, it is the tendency of tablet or to powder, chip and fragment it can be determined. Under this fragmented test, the tablets, they are continuously moving under certain rotation, and we are checking how much material get uh, removed from the tablet. Now it is measured in the probability percentage loss. The formula is given here. The percentage loss is calculated by measuring the weight of the 20 tablet and minus the weight of 20 tablet after 100 rotation per 100. Now this difference gives the percentage of loss, and from that we can conclude if the probability is less than 0.5 to 1%, then the batch is acceptable. If it is more than 1%, then this batch is not acceptable. The material get again recycled. Now, in this case, the thickness and other leakages, the tablet strip is kept into the water or solvent, and we observed that after a certain time, as per the pharmacopoeia, the strips are removed, and then after removal of the tablet from the strip, then we are checking whether the solvent like water is entered into the tablet or this. In this way, in the second part, we look towards the all disintegration test, dissolution test, difference in that, and then some test, basic requirement in the pharmaceutical industry. Thank you.